Hello student, myself Usharani, biology lecturer, VMC PU College, Chitradurga. So today we are going to discuss about the first PU chapter, that is biomolecules. What is meant by biomolecules? So these are the organic compounds which are essential for all the living organisms. So these are some total of different types of biomolecules, compound and ions present in the cell is called as cellular pool. The biomolecules are the compound of carbon. Hence the chemistry of living organism is organized around the carbon. Carbon is the most versatile and most predominant element of life. So there are different types of elements, that is like hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, silicon, etc. So the biomolecules are classified into inorganic and organic molecule. The inorganic molecules involves minerals, gases and water, whereas the organic it involves carbohydrates, lipids, amino acids, proteins, enzymes, nucleotides, nucleic acids and even vitamins. So the biomolecules are classified into two forms like biomicromolecules and biomacromolecules. So the name itself describing that. So those biomolecules that having an less molecular weight, they are called as micromolecules. For to analyze this, the living tissue must be grinded with the help of pestle and mortar by adding an trichloroacetic acid. So those molecules that dissolved in acidic media, so they are present in acid soluble pool, and those does not dissolve, they are present in the form of pellet. They are called as acid insoluble pellet. So here the biomicromolecules, they are smaller in size, they are having low molecular weight, that is from between 18 to 800 daltons. They are found in acid soluble pool. So for that minerals, gases, water, sugar, amino acids and nucleotides. Whereas the macromolecules, they are large sized, they are having an high molecular weight above 10,000 daltons, found in acid insoluble pool. So example, carbohydrates, lipids, proteins and nucleic acid. So the first biomolecule, that is a carbohydrate. So what is meant by a carbohydrate? They are the hydrates of carbon. This is a simple definition. So even they are the polyhydroxyaldehyde or ketone groups. So they are present in most abundant organic molecules in the nature. The term carbohydrate is derived from the French term, that is anhydrate decarboné. So that is the empirical formula of carbohydrate is CnH2ON. So they are present in the ratio of 1:2:1. Functions of carbohydrates. So these are most abundant source of energy. They are the precursor for many organic compounds, and even they are present as glycoproteins, glycolipids in the cell. So they are present as a structural component in cellulose in the plant and even in the exoskeleton of some insects, and even the cell wall of microorganism, the storage form of energy to meet the energy demands of the body. So the carbohydrates are classified into monosaccharides, oligosaccharides, polysaccharides. Monosaccharides. Here carbohydrates are commonly called as saccharides, nothing but sugars. So monosaccharides, name itself describing that mono, nothing but single. Here this sugar, it has been formed by a single unit. There is a basic unit of carbohydrate. They cannot be hydrolyzed into smaller unit. Based upon the number of carbon atom and the functional group, they have been classified further. Another one, that is oligosaccharides. Oligo means few. They are having a few monosaccharide or monomer units. So they can be further hydrolyzed. They consisting of disaccharides, trisaccharides and tetrasaccharides. Next one, that is a polysaccharides. Poly, nothing but many. So here the saccharides more than 10 or 9 in number. So here these are non-crystalline, non-soluble in water. They are tasteless. On hydrolysis gives molecules of monosaccharides, example starch and cellulose. So monosaccharides, based on the number of carbon atom, they are classified into trioses, tetroses, pentoses, hexoses and heptoses. So the example for trioses, that is glyceraldehyde, for tetroses, erythrose, pentoses, ribose, ribulose and deoxyribose. Ribose and deoxyribose, which are present in these are the pentose sugar present in DNA and RNA. Hexose, example glucose, fructose. Heptoses, that is pseudo-heptulose. Based on the functional group also, the monosaccharides have been classified, that involves aldoses and ketoses. So the derivatives of monosaccharides, they are derived in the form of deoxy sugars, amino sugars, sugar acid, sugar, alcohol. Oligosaccharides, here these are formed by the condensation of 2 to 9 monosaccharide unit. Depending on the number of monosaccharide, they are classified into disaccharides, trisaccharides, tetrasaccharide. The smallest and commonest oligosaccharide, that is called as disaccharide. So disaccharide, they consisting of 2 monosaccharide units, they are held together by the help of glycosidic bond. So these are crystalline in structure and they are water soluble, they are sweet to taste. So example for disaccharide, that is maltose, lactose and sucrose. Maltose is also called as malt sugar, they are made up of 2 glucose molecules. Lactose is also called as milk sugar as it has been found naturally in the milk. It has been made up of glucose and galactose. Another one, that is a sucrose, which is called as cane sugar, so which has been found in sugarcane and sugar beet. Most abundant among naturally occurring sugar. So it is most important dietary source also. So it has been made up of glucose and fructose. So another carbohydrate, that is called as polysaccharide. So it has been made up of more than 9 number of monomer units. So they are also called as glycans. They are made up of an repeating unit of monosaccharides and they are held together by the help of glycosidic bonds. So during its formation, a water molecule is released at condensation. It helps reduce the bulk, making it almost insoluble, decreasing its effect on the water potential and osmotic potential of the cell. Unlike sugar, they are not sweet. So they are ideal as storage and structural component. They are of 2 types, homoglycans and heteroglycans. Even they are called as homopolysaccharides and heteropolysaccharides. The homopolysaccharides or homoglycans, they are made up of only one type of monosaccharide, monomers. For example, starch, glycogen, cellulose. Whereas heteroglycans or heteropolysaccharide, they are made up of by the condensation of 2 or more type of monosaccharide. Example, agar, chitin, peptidoglycans, hyaluronic acid, etc. So here the polysaccharides which are present in the storage form, that is a storage polysaccharide. First one, starch, which is commonly called as reserve food material in plants. So even it is a most dietary source for all the animals. High content of starch, it was present in cereals, roots, tubers, vegetables, etc. So the homopolymer, it has been made up of glucose, also called as glucon. The starch, it has been formed by the combination of amylose and amylopectin. Glycogen, which is called as reserve food material in animals, that is hence they are referred as an animal starch. It was present in higher concentration in liver, muscle and even brain. It is also found in plant that do not have chlorophyll. So glucose is the repeating unit. So another storage polysaccharide, that is inulin. It is a polymer of fructose. It was found in dahlia bulbs, garlic, onion, etc. So they are easily soluble in water. Inulin is not readily metabolized in the human body and is readily filtered through the kidney, hence used for testing the kidney function. So structural polysaccharide, it involves cellulose and chitin. So cellulose, it occurs extensively in plants and it is most abundant organic substance in plant kingdom. So predominantly it is helpful for the constituent of an cell wall in plants. It is totally absent in animals. Chitin, it is the second most abundant organic substance. So where it has been formed by a heteropolysaccharide. So it was found in the exoskeleton of some of the insects like and even crustaceans. Provides both strength and even elasticity. So that becomes hard when impregnated with calcium carbonate. So another biomolecule, that is a protein. So proteins are the polypeptide chains of an amino acid. So the proteins are formed by an basic unit, that is a building blocks called as amino acids. So here these are the most abundant organic molecule of a living system. They form about 50% of the dry weight of the cell. They are most important for the architecture and functioning of the cell. So proteins, it has been formed by the polymers of an amino acid. So there are 20 amino acids are present. So in that the collagen, it is a protein that was present most abundant in an animal world. In biosphere riboscope, so that protein was present in an excess amount. It has been abbreviated as ribulose biphosphate carboxylase oxygenase. Even protein synthesis is controlled by DNA. So the basic unit of proteins, they are called as amino acid. So these are the organic compound which consisting of amino and carboxylic group. So the amino, it has been denoted by NH2, whereas carboxyl with COH. So in the right side of the slide, so we can observe the structure of amino acid, where the carbon, it has been constituted or arranged along with amino group and even carboxylic group along with R group. R sometime it may varies. So R can be H group in glycine, CH3 in alanine, hydroxymethyl in serine. In others, it can be hydrocarbon chain or a cyclic group. All amino acid contain C, H, O and N, but some of them additionally they contain S. Physical and chemical properties of amino acids are due to the presence of amino and even the carboxylic group and even a R group. Amino acids are differentiated into 7 groups. These are as follows. There is a neutral. So here the amino acid which was showing a neutral property, for example a glycine, alanine. So those amino acids which was showing an acidic property called aspartic acid, basic arginine, S containing amino acid, example cysteine, alcoholic amino acid, example serine, aromatic, so which is example phenylalanine, heterocyclic, so example histidine. So based upon the amount of amino and carboxylic group, the amino acid it was showing both acidic and basic property. At particular pH, both amino and carboxylic group, they get ionized. So that can be defined as an Zwitter ion. So here the amino acid, it was showing an amphoteric in nature. Nothing but it can react with both acid and basic. So here the peptide formation. So the amino acid, it has been combined or joined together by a linkage or bond called as peptide bond. So in right side we can observe that one amino acid, second amino acid, it has been linked by the help of peptide linkage. So it has been formed by the carboxylic group of first amino acid and amino group of second amino acid. So the chain having a 2 amino acid, they are linked by the peptide bond is called as a dipeptide bond. Here the sequence of amino acid present in the polypeptide is specific for the particular protein. Structure of protein. So protein based on the structure have been classified into 4 types. There is a primary, secondary, tertiary and quaternary. So here the primary structure, it refers to, here the amino acids of an polypeptide, it has been linearly arranged one after the other, in that the N-terminal amino acid is written on the left side, whereas C-terminal amino acid is written on the right side. In secondary structure, so here the linear chain, it has been showing an coil-like structure that to form an different types like alpha helix, beta pleated sheet and collagen fibers. In tertiary structure, so here the polypeptide chain, it has been super coiled together to form an halo woolen ball like structure. So where the functional amino acid has been exposed outside. In quaternary structure, it has been made up of 2 or more polypeptide chain. Example for quaternary structure amino acid, that is called hemoglobin. It has been formed by 2 alpha chain and 2 beta chain. So there is a photocopy, there is a 3-dimensional photocopy of an structure of protein. So in first, that the primary structure, so where the amino acid have been linearly arranged. In secondary structure, they have been spirally coiled together. So in tertiary structure, so it forms and spirally coiled together in like an hollow woolen ball like structure. So in a quaternary structure, it has been formed by more than 2 polypeptide chain. So proteins. So in that, the functional property, chemical nature and nutritional importance can be studied further. The functional classification of protein. So it is a structural protein, example keratin, collagen. So some of the proteins that acts like an enzyme, that is an enzymatic protein, example pepsin. Transport protein, example hemoglobin. Hormonal protein, example insulin and even a growth hormone. Contractile protein, actin and myosin. Storage protein involves ovalbumin. Genetic protein, that is a nucleoprotein. Defense protein, it involves immunoglobulins like antibodies. Receptor proteins for hormones and viruses. So classification based on chemical nature and solubility. So here are classified into simple proteins, conjugated proteins and derived proteins. So simple proteins, they are composed only of amino acid residues. Conjugated, along with amino acid, there is a non-protein prosthetic group is also present. In derived proteins, they are denatured or degraded product of above 2. So proteins, in that the simple, conjugated and derived proteins. The example for globular protein, there is an albumin, globulin and histone. So here the simple proteins are classified into globular and fibrous. The globular protein, it involves albumin, globulin, histones, whereas the fibrous, it involves collagen, elastin and keratin. The conjugated proteins, it involves nucleoprotein, glycoprotein, mucoprotein, lipoprotein, phosphoprotein, chromoprotein, metalloprotein. So whereas the derived proteins, it involves primary and secondary. In primary, coagulated protein, proteons and metaproteins. In secondary, proteoses, proteins and peptides. So next one is lipids. So lipids are the chief concentrated source from the energy form. They are present in the form of 3.5% of the cell content. So lipids are the organic substance. Relatively, they are insoluble in water, but they are soluble in organic solvents like alcohol and ether. So the functional property of lipids. They are the concentrated fuel reserve of the body, and lipids are the constituent of membrane structure and they regulate the membrane permeability. They serve as a source of fat soluble vitamins. Even the lipids are important cellular metabolic regulators. So here the lipids protect the internal organs and serves as an insulating material. Lipids are classified into simple, complex and derived. The simple lipids, it involves fats and oils, waxes. The complex, it involves phospholipids, glycolipids and lipolipids. Derived, it involves steroids, terpenes. The simple lipids. So these are the esters of fatty acids with alcohol. So there are of two types. There is a neutral and waxes. So the neutral fats, they are the esters of fatty acids with glycerol. Whereas the waxes, they are the esters of fatty acids with alcohol other than glycerol. So neutral fats. So they are the true fats. They are made up of C, H and O, but O is less in amount. A fat molecule is made up of two components like glycerol and fatty acids. So glycerol, it is a molecule have 3 carbon each bearing OH group. So fatty acid, it is a molecule is an unbranched chain of C atom. It has COH group at one end and H bonded to almost all the C atom. Fatty acids may be saturated and unsaturated. So based on the specific structural component. So there are essential fatty acids and non-essential fatty acids have been classified. Essential fatty acids, that is abbreviated as EFA. So there are 3 essential fatty acids are required for our body, that is called arachidonic acid, linoleic acid and linoleic acid. So next one, that is a waxes. Lipids which are having a long chain structure, saturated fatty acid and long chain saturated alcohol of higher molecular weight other than glycerol. So example, beeswax, wool fat, sebum, cerumen, plant wax and paraffin wax. Complex lipids. So these are the derivatives of simple lipids having additional group like phosphates, N2 bases and protein. So they are further divided into phospholipids, glycolipids and lipolipids. So the first one, in diagrammatic photocopy, we can observe the structure of phospholipids, so which consisting of an head and tail region. So head, it was showing an polar property, that is it is hydrophilic in nature. So whereas the tail, it is an hydrophobic in nature. So which has been formed present in an cell membrane in the form of biphospholipid layers. Glycolipids. So these are the component cell membrane, particularly myelin sheath and chloroplast membrane. So derived lipids. So they are obtained on the hydrolysis of simple and complex lipids. Example, sterols, cholesterol and ergosterol. So the major complex biomolecules of cell. So it involves protein, DNA, RNA, polysaccharides and lipids. So the proteins, it has been formed by a building blocks called as amino acid, DNA with deoxyribonucleotide, RNA with ribonucleotide, polysaccharide with monosaccharide, the lipids with fatty acids and glycerol. So next biomacromolecule, that is nucleic acid. So these are the molecule that has been formed by a nucleoprotein. So where there are two types of nucleic acid, that is DNA and RNA, called as deoxyribonucleic acid and ribonucleic acid. So it has been formed by the units called as nitrogen bases, that is purine and pyrimidine. So in purine, adenine and guanine, in pyrimidine, cytosine and thymine. So whereas thymine is replaced by uracil in RNA. So another basic component in nucleic acid, that is called pentose sugar. When this nitrogen bases are combined with a pentose sugar, so it forms nucleosides. When these nucleosides combined with the phosphate groups, it forms an nucleotides. So nucleotides are the basic units or the building blocks of a nucleic acid. So where the nucleic acid can also be defined as there are the polynucleotide chains. So here, the structure of DNA. The structure of DNA has been described by scientists called as James D. Watson and Francis H.C. Crick. So in this, the double structure of DNA, where this each strand, they are running in anti-parallel direction, they are having an sugar and phosphate which acts like an backbone. So where in this, they are showing an complementary base pairing, where adenine always base pair with thymine with 2 hydrogen bonds, guanine base pairs with cytosine with 3 hydrogen bonds. And the amount of purines is always equal to the amount of pyrimidines, which follows an Chergaff's rule. So here, the one turn of DNA, it has been constitute 10 base pairs, and they are showing a distance of about 3.4 angstrom, and each are separated by 0.34 angstrom. So next one is enzymes. So the enzymes are a group of catalysts. Their functions as an biological system. So the study of enzymes is called as enzymology. So the enzymes occurs in colloidal state and they are often produced in an inactive form. They are called as proenzymes or zymogen. So the enzymes that are produced within a cell or metabolic activities are known as endoenzymes, and some which act away from the site of synthesis are called as hexoenzymes. So enzyme action has been predicted. Here enzymes are like the structure like protein, which involves primary, secondary, tertiary structure. So during spirally bounding, it was showing an active site. So towards the active site, another chemical compound which is helpful for the formation of product, it combines to the enzyme called as substrate. So in photocopy representation, the enzyme, so at a particular active site, the substrate it binds that to form an enzyme substrate complex. After the formation, so it was producing the products by the help of substrate. Again the enzyme become free that to combine with another substrate that to show an catalytic cycle. So here a reaction, the enzyme when it combined with the substrate at an active site, it forms an enzyme substrate complex, that is enzyme bind with the substrate. After this, the enzyme it releases the products. So whereas in inhibition, instead of substrate, when the enzyme has been combined with the inhibitors. So there is no synthesis of an product where it inhibits the compete with the substrate. So this is the graphical representation that to describe the activation of energy. So enzyme is necessary for to increase the activity of an chemical reaction. So where if the enzymes are present or absent, it was showing an exothermic and endothermic process. So where activation of energy with the help of enzyme, it was showing a less initial process. Without the enzyme, it has been showing an higher peak level. So that means for the progress of reaction, so energy, it has been completely depend upon the presence or absence of enzyme. So the general property of enzymes. The enzymes have been accelerated the reaction but do not initiate it. So enzymes themselves do not participate in the reaction and they remain unchanged at the end of the reaction. So the molecules of enzyme is larger than that of the substrate and they're having a specific site called as active site. So here it was showing amphoteric nature. Chemically, most of the enzymes are proteins and therefore show amphoteric nature. The enzyme can react with acidic substance as well as alkaline substance. Specificity. Most of the enzymes are specific in their action. A single enzyme acts upon a single substrate or group of closely related substrate. For example, the enzyme urease can act only upon urea invertase, can act upon sucrose only. So another one, that is a colloidal nature. All enzymes are colloidal in nature and thus provides the large surface area for reaction to take place. Colloidal are mixtures of two components, that is dispersed particles and dispersion medium. So enzyme optima. Enzymes generally work best under certain narrowly defined condition. So they are referred to as optima. So these includes appropriate temperature and even pH. So enzymes are temperature sensitivity. So here, since the enzymes are proteins, they are affected by the change in the temperature. In higher degree of temperature, the enzymes become denatured. At low temperature, they become inactive. So they are pH sensitive. Most of the enzymes are specific to pH. So they remain active within a particular range of pH. The strong acidic or strong base denatures the enzyme. Most of the intracellular enzymes functions best around the neutral pH. The concentration of enzyme and substrate. The rate of reaction is proportionate to the concentration of reacting molecule. If the substrate concentration is increased, the rate of enzyme action also increases. So enzyme inhibitors. So these are the certain products which inhibits the enzyme activity. So during the reaction, if the active site of an enzyme is occupied by these inhibitors instead of substrate molecules and the activity of enzyme is lost, these substances are like substrate molecule in their structure and they are called competitive inhibitors. So the group of enzyme based upon the functional property, they have been classified into different forms like oxidoreductases, transferases, hydrolases, isomerases, lyases and ligases. Oxidoreductase, they are helpful for the transfer of oxygen and even the hydrogen atom. Transferase is helpful for transforming the specific group from one substrate to another, whereas hydrolase helpful for hydrolysis of substrate. Isomerase helpful for the change of molecular form of the substrate that can be interconvertible. Lyases, so helpful for non-hydrolytic removal or additional of a group to the substrate. Ligases are helpful for joining of two molecules and to form a new bonds. So let us discuss about some of the MCQs in this chapter. That is, which of the following is not strictly a biomacromolecule? So macromolecules nothing but those organic compounds having an higher molecular weight, that is more than 10,000 daltons. So here the given option, that is protein, lipids, polysaccharides, nucleic acid. So in this, the proteins, polysaccharides and nucleic acids, they are the macromolecules. But the lipids, they are not completely considered macromolecules because the molecular weight of lipid does not exceed 800 daltons, but they come under the acidic insoluble fraction. So the correct option for this question is B, lipids. In glycine, the R group is replaced by. So here, glycine it is an amino acid. So where it has been, when it consisting of R group in an amino acid, it can be a methyl group, hydroxyl methyl, carboxylic group, D option, hydrogen. So here the correct option for this, the glycine is the simplest amino acid with an R group. It has been replaced by the hydrogen. So the correct option for this question is D. In glycine, the R group is replaced by hydrogen. Zwitter ion. Here at particular pH, so both amino and carboxylic group in an amino acid, they get ionized. That is called as Zwitter ion. The given option, that is an acidic amino acid, basic amino acid, neutral amino acid, all of these. So here it can showing an at particular pH it get ionized. So the given option, correct option for this is D, that is all of these. Since amino acid can carry both positive and negative charges simultaneously, hence they are termed as an Zwitter ions. So fourth question, that is a non-essential amino acid. So here the amino acids are classified into essential and non-essential among 20 amino acids. Essential amino acid nothing but the amino acids that are not synthesized in our body. They must be taken through the diet. But whereas non-essential amino acids they are not synthesized in our body, they must be taken through the diet. Whereas non-essential, they are already synthesized in our body, they need not to be taken through the diet. So in that, the given option must be obtained from the food or synthesized in our body or not needed in our diet. Option B and C, both B and C. So here must be obtained from the food. Those are the amino acids are essential. Here the non-essential or synthesized in our body, they are need not to be taken through the diet. So the correct option is both B and C. Option correct is D. So the most abundant protein in the animal world. So the given option, chitin, collagen, peptidoglycan and hyaluronic acid. So in that, the protein that was present most abundant in the animal world is collagen. So the correct option for this is B. So whereas chitin, peptidoglycan and hyaluronic acids, they are not a protein, they are the polysaccharides. The amino acid in a protein are held together by. So here the given option, glycosidic bond, phosphodiester bond, peptide bond and hydrogen bond. So here the glycosidic bond, it is a bond that has been formed between a monosaccharide, whereas the phosphodiester has been formed between the nucleotides, that is during the formation of DNA. So whereas the peptide bond has been formed between the amino acid. Hydrogen bond, it is also specific during the formation of an primary and secondary structure of an protein. So here the amino acid have been linked together by the help of peptide bond. That is the correct option, that is a C. So which of the following are the basic amino acids? So the given option, glycine and alanine, lysine and arginine, glutamic acid and aspartic acid, histidine and proline. So here lysine and arginine are the basic amino acid, they are positively charged. Glutamic acid and aspartic acid, they are acidic amino acid, they are negatively charged. So the correct option for this is B, that is lysine and arginine. A nucleotide is made up of. So here the nucleotides, they are the basic unit of an DNA and even RNA, have been formed by the combination of, there is a nitrogen base plus pentose sugar plus phosphate. So let us discuss about the option. Base, there is a nitrogen base, sugar, phosphate, there is a N, nitrogenous base and sugar, nitrogenous base, sugar and phosphate. D option, phosphate and N base. So here the correct option for 8th question, it has been formed by the combination of nitrogen base plus sugar, that is a deoxyribose or ribose sugar, and phosphate. So the correct option for 8th question is C. So which of the following is reducing sugar? So here the option galactose, gluconic acid, methyl galactoside and sucrose. Among these, the reducing sugar, that is called as an galactose. So the correct option for this is A. So a competitive inhibitors of succinic dehydrogenase. A competitive inhibitor having a structure that they are similar to the substrate that inhibits the chemical reaction. So the given option, that is a malate, malanoate, oxaloacetate, option B and C. So the correct option here, these are the competitive inhibitors of succinic dehydrogenase or malate, malanoate and oxaloacetate. So the correct option for this is B and C. That is a D option is correct. Which of the following has a carbohydrate as a prosthetic group? So here, first option, glycoprotein, chromoprotein, lipoprotein, nucleoprotein. So here in this, it is a conjugated protein in which the protein is conjugated with a carbohydrate. That is the correct option is A. An organic substance bound to an enzyme and essential for its activity is called as. Here, the organic compound which always bind with an enzyme, either coenzymes or prosthetic group, the given option isoenzyme, coenzyme, holoenzyme, apoenzyme. So the correct option for this question is coenzyme which bound to the enzyme. Lipids that insulate the nerve fiber. Option lecithin, cholesterol, suberin, glycolipids. So here glycolipids are present in myelin sheath of nerve cell. So for this, the correct option, that is a D, glycolipids. Another one, that is a nucleosides. So it has been formed by the addition of an nitrogen base plus pentose sugar. So let us discuss about the option, that is a sugar plus nitrogen base, sugar plus phosphate, nitrogen base plus phosphate, purine plus pyrimidine. The correct option for this is sugar, that is a pentose sugar plus nitrogen base. So when this has been added with a phosphate, it forms a nucleotide. So the correct option for nucleoside is A, sugar plus nitrogen base. Metabolic flux is regulated by. Flux nothing but the movement. When the molecules they are moving inside, that is called as influx. When move outside, it is called as efflux. So here, the metabolic flux is regulated by enzymes, sugars, phospholipids, sterols. Here, the metabolic flux is the rate of turnover, that is the total chemical reaction, it is called as a turnover of the molecules through the metabolic pathway. It has been taken over by the help of a component called as enzymes. So the correct option for 16th question is enzymes. So these are all about MCQs in this chapter. So okay, thank you students.